exchange and returns would be our nucleus in relation to target, because behind these walls we have the inventory for the whole store, so that would kind of replicate the DNA storage and how that controls everything, and the inventory, you could say, controls target. And the wall of the exchange and returns would represent the nuclear envelope, or the membrane that surrounds the nucleus.
And the exchanges and returns serve as our nucleolus because they hold all the information for all the inventory in target, which is similar to how the nucleolus holds all the information for everything in the cell.
The smooth endoplasmic reticulum is responsible for creating lysosomes, steroids, and also getting rid of the cell's toxins. And for our example, we went into the liquor aisle, as the smooth ER makes up most of our liver, and our liver's main job is getting rid of this stuff. In continuance, the structure of the smooth endoplasmic reticulum is very similar to that of the rough ER, except for it lacks any ribosomes, so our representation still stays the same, as there is no definitive sign that stands over our smooth ER in target.
The rough endoplasmic reticulum is responsible for the synthesis and packaging of proteins in the cell. It also has ribosomes on the outside, which make it definitive from all the other organelles in the cell. And so we chose the CAFE to embody our rough endoplasmic reticulum because, as it's ribosomes, it has a sign that says CAFE up at the top, and it is also responsible for the making and packaging of different foods and stuff for the customers in Target.
And up next we have the Golgi apparatus, which we are using for the CVS pharmacy attached to Target. And here the Golgi modifies and distributes proteins out to other parts of the cell. So our connection back to the pharmacy is how they modify and distribute prescriptions out to customers. And as for the structure, we found it similar in the Golgi apparatus's like folds and membranous sacs to the aisles. We saw that it was cis, trans, and medial in the way that it was looped all together. And now we have arrived at the mitochondria, which is also coined the powerhouse of the cell. And we used a back supply closet to represent this, as just like the mitochondria generates ATP, or energy, we tied the analogy to the supply closet holding and generating stock and inventory for the store. As for structure, we found the membrane being very similar to these flimsy doors, and if you look inside, they also have aisles in there, similar to that of the Golgi apparatus, is located in the folds of the inside of a mitochondria.
Chloroplasts in a plant cell take energy from the sun and they turn it into food, which is used as energy for the plant cell. Much like lights take electricity from batteries or any power source and they turn it into light that we use to see. Structurally, chloroplasts have different compartments and they are found in different areas of the plant cell. Much like these lamps have little compartments that you could stick other stuff in, and you could even plug in other stuff to make more energy and power. Vacuoles in the cell are responsible for holding water, and they take up most of the space within a cell, and is modeled by our little tunnel that we have, which is blue, and it goes like this.
And now we have lysosomes, which are responsible for digestion and waste removal in the cell, and we of course compared that to a restroom. So as for the structure, they are membrane-bound organelles that contain a whole bunch of enzymes in them that aid in the decomposition of waste within a cell. And we compare that to the stalls of a restroom being like the membrane-bound, and then the toilet or toilet paper or anything else would represent the enzymes that help with digestion. And now we have the cytoskeleton of the cell, and we have the analogy to a beam in Target. So the cytoskeleton acts to help maintain the shape and structure of the cell, just as this beam helps keep the roof intact and maintain the shape and structure of the building itself. The cytoskeleton is represented by filaments located within the cytoplasm, and that is very similar to how this beam would look compared to the floor and intact with the roof. And now we have centrioles, which are located in animal cells and aid in cell division. So for our analogy, we have the centriole represented by this pool noodle, and it's basically just a cylindrical molecule. So that's that. And for the chromosomes, which the centriole aids in splitting during cell division, are represented by these two pool noodles. So now we shall reenact what a centriole does within the cell. And there you got it.
We have chosen the outside of target to represent the plasma cell membrane because, like the plasma cell membrane, it is the outside of the store and it is protective of the cell and its contents on the inside. And the plasma cell membrane is a phospholipid bilayer, kind of like how all the walls are on the outside and they protect the inside of the store. We have decided to vote off vacuoles from our island because
we feel that there would be another way that cells would work around getting water into the necessary nutrients that they need. And the vacuoles take up the most space in the cell, so we believe that those could be filled with other members of the endomembrane system that could better aid production of proteins or any sort of thing within the cell. So vacuoles could go, find a new source of water or new way to filter in water, and they're off our island.